So without further ado, I'll hand over to the first of our speakers in what I think is going to be a very interesting and important session on governing genomic data. Lucy.
So I'd like to thank the organisers for the invitation to talk about the UK 10K, which is a UK-based project which was launched in 2010. It's a Wellcome Trust strategic award running over 3 years. And I will talk about, I was part of the governance committee. So first, because 2010 in some ways for genomic research seems quite a long time ago, I'm going to talk a little bit about the background and the context from which the UK 10K emerged. Talk about the principles governing the UK 10K, which we devised as we, didn't make it up as we went along, but certainly we started and then had to create a framework fairly rapidly. And then I'll do a sort of short reflection on where we are in 2014, because I think the landscape is now quite different. The context to this is the 1,000 Genome Project, for which this is a huge international effort, of which the Sanger Institute was a major player. And the aim of the 1,000 Genome Project was to identify common variants in the human genome and to make these freely available for the research community as in the widest possible way it could. Now, critically, in that in the 1,000 Genome Project, it took 2,500 unidentified people from about 25 populations and performed next-generation sequencing on that to identify the remaining common variants in the population. And these could be publicly available, easily accessed, and available to any of those who want to use it for subsequent studies. The aim at that time was to do a relatively modest coverage of only about 6-fold, so actually you couldn't, the depth, you would only really reveal the most common variants. And this was launched in 2008. And just as it was beginning to report, it became clear that actually the depth of coverage was too low to be useful to identify the more intermediate and rare variants. So the UK 10K project was emerged from that stable, and that was the principle. It was a UK-based population. And the primary aim was to increase the coverage of data throughout the genome, both to identify the frequency of intermediate alleles and in particular to start really looking at rare diseases. And in particular was to look at these low frequency alleles and in particular to think about strategies for looking at rare diseases. And so one of the specific briefs of the UK 10K was to look at a cohort of patients with rare diseases to identify high penetrant rare alleles. And in true Wellcome Trust style, this was absolutely intended to be available to the wider research community on a non-profit making basis. So the 10,000 individuals actually formed very discrete two groups. One, 4,000 individuals for whom whole genome sequencing was going to be performed, still at relatively low resolution compared to now, but certainly better than the 1,000 Genome Project. And that cohort came from these volunteer well communities such as the ALSPAC and the Twins UK cohort. And then there were 6,000 of these individuals who were going to have whole exome analysis done, and they had rare diseases. And there were a range of rare diseases from neuropsychiatric disorders to some even very rare diseases such as collarbone ciliopathies, and the list is there. And also there was a replication study as well included in that cohort. So the whole study design has incredible intrinsic tensions to it. And that became very obvious at the very beginning of when we were setting up the study. There's a desire to make research data available to the wide research community, and that was very much the stable from the 1,000 Genome. Large-scale anonymous data sets are incredibly valuable in terms of interpreting data. But we were trying to look at rare variants that cause a rare disease. And we know that having a rare variant in an individual is quite sufficient to be able to identify who that person is. And the other thing is that most of these rare patients, or patients with rare disease, I should say more correctly, were coming from a context of being already investigated within the NHS, where the NHS had exhausted the available. So the tradition for these was about patient confidentiality. And there was an incredibly clear need to protect patient confidentiality in the context of this clinical research setting. So those two contrasts, and the image that I find very useful in thinking about this is the Anthony Gormley, who's a famous UK sculptor who looked and populated large shed of multiple different peoples, and you begin over looking at large data sets, you get most fantastic trends and shapes. But of course, you need to also remember that these are all individuals. And to me, those are the two tensions in this whole project. So we found a solution because we needed to. And at that time, there was increasing literature illustrating that the identifiable nature of people based on just looking at a panel of common SNPs, let alone rare SNPs. And all you need is one additional sample, albeit you do need an additional sample. The data itself doesn't identify them, but the simple process of having a single sample from an individual that does have identifiable data, and the triangulation point means that you effectively impute the whole genome. So on the basis of that, we adopted what we called a managed access system where data was available, but only to bona fide researchers who had to justify their reasons for having access to that data. We made that as loose as possible, exactly comparable with the WT-CCC cohort. And the minimum criteria we said was that they had not to try and identify who the patient was. And they also needed to be generating peer-reviewed journal data so that we were clear that they were bona fide researchers, but we didn't need to make much more judgment than that. And that the PI it's only PIs who could apply for access, and the PI undertook responsibility of management of that data on behalf of all of their staff. So this was not just a feeding frenzy of anybody, it had to be very quite carefully controlled. And whilst we couldn't guarantee privacy, at least we were setting up some appropriate mechanism to protect potential participants from exploitation. So having set that scene, it was important that we then started to think about an ethical governance framework for this cohort. And the particularly challenging was the rare cohort. The rare cohort was made up of multiple different cohorts that had already been consented for research. And they were already within an existing REC, so they were ethically approved studies. However, we found that many and most of them needed to be reconsented. And the single thing that was probably the most common reason why I needed to reconsent was that previous ethics had almost never addressed the issue of data access and data in perpetuity, because we didn't really think about it in the noughties. We just didn't. We just assumed that projects were finite. And the concept of having data sets which were available in perpetuity. Also, our RECs, some RECs, and interesting, not all, but a few RECs were quite concerned just simply about the volume of data. Many of these rare studies had previously gone to candidate genes, they'd gone to systematic arrays, and suddenly we NIGHTS moved into exomes and genomes. And there was a concern of just sheer volume of data. There wasn't any qualitative difference, but it was just the sheer volume of data. And it was felt that patients should be reconsented to be aware that that was the case. In our experience for most of the rare diseases patients that we had already recruited, consent was usually implicit in the consent was that almost all the studies had an option to receive feedback, and every single one had ticked that. It was almost a mandate, because essentially that was often the main reason why people were being recruited into a research study. And the overwhelming motivation, if you have a rare disease, is to actually find the cause of your disease. So it was pretty obvious that most people actually wanted feedback. However, there was also a high degree of altruism amongst this group. So many of these people realized that they may not get feedback, but they were very happy to be part of a study which would benefit others. And certainly that's based on the work that we did on the GOLD study, where we interviewed families about what they felt about being taken part of research. And so what it raised was, if we now do whole exome data, what is the duty of care for the research? Obviously, now we are very familiar with the concept of pertinent findings and incidental findings. At the time, we, again, had a lot of rhetoric about what names to call it, but essentially we talked about pertinent findings, and we decided not to feed back incidental findings at all. And that was partly a long debate that we had amongst ourselves. Now, most of the PIs for the rare diseases were clinician scientists, so they have feet in both camps. They were providing clinical care for these patients, and they were recruiting patients for their own research studies. And so initially, when we all talked about it, we were massively enthusiastic about reporting incidental findings. This was great. We would do a duty of our care to our patients, until we started thinking more profoundly about that. Now, every PI had international expertise in their area of research, but they would be the first to admit they had no experience in anybody else's area of excellence. And so the questions that came up and the themes was, what is our competence as clinician scientists to interpret the data on genes we know nothing about? which genes should we be looking at, and more particularly which variants in which genes, because not all variants are equally penetrant, and just because they're rare doesn't mean they're pathogenic. What degree of validation are we going to have to do on genes that we know nothing about? And really, where is the evidence of penetrance? What's the likelihood of this actually affecting somebody in an unaffected population? It also then raised the issue about what is our duty of care as clinicians to seek out potentially hazardous variants opposed to just truly incidental. And there was also a very practical reality of what is the source implication of doing this when, we actually couldn't afford to do it under the current framework, but actually I think it's important to realise that actually it was much more a scientific concern about our medical ignorance to interpret this data was very much the overriding concern from our point of view. So having agreed that we would only feed back clinically significant findings, we needed to devise a framework by which we wanted to judge that was the case, because what we did feel very clearly was that it was not justified for a research project just to dump all of this data onto a clinical service saying, go find, go verify. That seemed unfair and unjust and inappropriate use of resources. So we had a very clear system that we would only feed back variants that we were very clear and confident that this was going to be the cause of disease or have a material effect on the disease for which the patient was recruited. And also we carried a responsibility to validate that within a research setting before we then handed it on to the clinical service. However, we also had a caveat that all reports that we sent had to be verified within a clinical service with a CPA-accredited laboratory because none of the research cohorts were ascertained on that basis. There are well-established, in the UK, well-established mechanisms of feeding back through clinic. Many research projects have gone for many years doing this very successfully. We didn't see any reason to change that. Sending a preliminary research report back to a clinician, going back to the patient, having a consultation, rebleeding, confirming the variant. And so we suggested that. And so that's what we did. So here we are in 2014. What did we do? Well, the UK 10K, I think, actually did achieve its aims. It now has over 35 published papers in the UK 10K and has made a significant impact on identifying new rare disease-causing genes. Now, I think if we'd spent our time looking at incidental findings, we would have maybe missed the brief that we had, and I think that's an important message. We have all only got limited resources, and actually our patient's brief was to find the cause of their disease because that's why they were recruited. Many of the rare variants that we've identified have been validated and been reported back to the participants, and certainly in my contribution, my cohort, we've got over 120 families now have the cause of their disease fed back to them and been validated. Nobody came back to us and said, we'd like to have our genome in our pocket, please. Nobody asked for incidental findings, and nor, fortunately, did we incidentally find any incidental findings. But we haven't had requests, and we've had no requests for the data from the participants. I think where I wouldn't say failed, but what's clear is that by having a managed data access system, that has been limited. There's been limited interest in having access to that. And of course, actually now, the Broad Institute have just dumped a most phenomenal data set, freely available to all of us, of 61,000 exomes freely just on the web. So I suspect the UK brief of being a data source that's going to be a controlled data source for many research projects may not be sustained because other projects have done that. In reflection, were we too cautious about data access? I think given the time in 2010, probably not. I think in new projects emerging, we were maybe slightly more liberal. I think what we have done is we've had no breach of confidentiality. We've not had any disasters. And because we got no news, I think that's actually quite a good message. We haven't had to manage situations where we've had confidentiality has been breached. And I think finally, we've used our resources maximally. I think we have to be sensible. Certainly Europe and the UK, we run a state healthcare system, and incidental findings is expensive. And they may outweigh the benefit of focusing on the reason why the people are recruited into the study. But that's a big debate, and I will feel that. So finally, I want to thank all the members that are part of the UK 10K, and in particular to Jane, who steered, was it 27 drafts of a paper, you said, because we all had our 3 penny worth. And thanks to the Wellcome Trust for the funding.